Hello, everybody. Welcome to this session again. We have a very special and very complicated paper today, and I hope Philippe and Ivar will manage. Yeah, I think that's it. Philippe, would you mind taking over the floor?
Sure. So, for the choice for today's paper is really because there are two main interests. So first, we are in the context of CPVT, and to start with, I'd like Ruben to show us what a CPVT is and explain us.
Do you see my screen?
Yes, we do.
So this is an ECG from a patient here at the AMC who has been diagnosed with CPVT, catecholaminergic polymorphic ventricular tachycardia. It's always a very complicated name. This patient was subjected to a exercise test, and at a certain moment, this was his ECG. You see the standard ECG leads, 1, 2, 3, FVR, AVL, AVF, and the precordial leads over here. And what you should notice is that there is a very rapid rhythm. I'm pointing here at V2 with a direction of the QRS complex upward here and downward there. So up, down, up, down, up, down. So it's called a bidirectional ventricular tachycardia. And of course, you can imagine that this is potentially very dangerous because the heart will not pump out enough blood. And these particular tachycardias are related to genetic abnormalities in the ryanodine receptor. And this is where the paper is about. So I'll stop sharing my screen and I'll give you the word back.
Okay. So the choice of the paper is really because for the first time in the literature, we have an interesting paper mixing the interaction of the mitochondrial compartment and the SR and another structure which we are studying into the metabolism group, which is the permeability transition pore. So just for you to know, this is a non-specific channel localized into the mitochondrial compartment. This channel is very well accepted in the literature for its participation in cell death, especially for infarct size development after ischemia reperfusion injury. But there's, for the last 10-15 years, some postulate that this channel would participate to calcium cycling and has a physiological role into the cardiomyocytes. For now, we don't know what that is. This is exactly the topic, the subject of KEPA's Master 2. We've been working on that since the very beginning. We had to stop these studies because we had the group has to focus on AF studies. But once we have students, M2 students or M1 students, we carry on this project and we have made good progress. So this is why we were very much interested into this paper in a good journal, even if we have some criticism, but it's a very good opportunity for us to let you discover the potential, the arrhythmogenic potential of the mitochondrial compartment into the cardiomyocytes. And now I would let the lead to Kepa Labesco, whom is in the 3rd years of medicine at the university and whom has done his M1 with us and now is doing his M2 and has made good progress into the comparison of the role of MPTP into mitochondrial calcium cycling. Enjoy the paper.
So hello everybody. I'm going to present you the paper called SR Mitochondria Crosstalk Shapes Calcium Signaling to Impact Pathophenotype in Disease Models Marked by Dysregulated Intracellular Calcium Release. This is a paper made by a few teams of researchers from United States of America. And this paper has been published in Cardiovascular Research in 2021. So first, I will make just a quick reminder, about the cardiac excitation contraction coupling. So in cardiomyocyte, when an action potential comes in the T-tubule, it triggers the calcium channel, voltage-dependent calcium channel type L, and this calcium entry triggers calcium exit from the sarcoplasmic reticulum, so the SR, through the rear channels. And this calcium is next used by the cardiomyocyte to contract the myofilaments. And during the diastole, so the relaxation, the calcium is pumped back 3 ways. One way through the CERCA ATPase pump, which pump back the calcium in the SR. There is also an entry of calcium in the mitochondria and an exit of calcium through the NCX pump, so the sodium calcium pump. And a bit more precisely about the mitochondrial calcium handling, you can see here the different channels working for the calcium handling of the mitochondria. First, the major entry pass of the calcium in the mitochondria is the MCU. So this is the mitochondrial calcium uniport. There is also a sodium calcium pump in the mitochondria, which make the calcium exit from the mitochondria. This is the main exit part of the calcium, but there is also the MPTP, so the mitochondrial permeability transition pore, which make calcium exit from the mitochondria. And so in this study, they are going to focus on 2 models, the CPVT2, so the catecholaminergic polymorphic ventricular tachycardia, and another model, which is the FFD, fast food diet mice. And why do they focus on those on those 2 models? It's because they have both enhanced DCR. So DCR stands for diastolic calcium release. I will explain you later what is DCR. And so those 2, DCR is diastolic calcium release from the rear channels during the diastole. And this calcium then is increased in the cytosol, which triggers a calcium wave, which triggers some arrhythmias. And so in those models, the CPVT type 2 is called type 2 because it's a mutation on the Casc2 gene, which encodes for the calcicrestin, which is a protein binding calcium in the SR. And when this protein is mutated, the calcium is not bind anymore in the SR, which triggers some DCR. And in the FFD, it's mice fed with some fast food diet, and this triggers a pre-diabetic cardiomyopathy, which is also characterized by a DCR. And so the question is in this paper is why, with both of those disease characterized by DCR, why this DCR triggers different effects in the cell. And so they are going to understand the different effects, and specifically by looking at the mitochondria role in those diseases. And so first here, this is a graphical abstract, so with the CPVT2 model and the fructose-fed model. And so they are going to focus on the entering of the calcium through the MCU and focusing on the exit of the calcium through the MPTP. And they are going to manipulate those flows of calcium through the campferol, which is an activator of the MCU, through the ruthenium 360, which is an inhibitor of the MCU, and thanks to also the CSA, which is an inhibitor of the MPTP, and they are also going to genetically inhibit those channels. So first, they were going to focus on the MCU with pharmacological inhibitors and activators. So they will take the mice heart, make a cardiomyocyte isolation, and load those cardiomyocytes with some fluorescent sensitive probes. So here you have an example with, firstly, they are going to use the camphorol. As I said before, this is an MCU activator, and the ruthenium-360, which is an MCU blocker inhibitor. And they are going to see the effect of those medicine. And in this example, they will firstly, here you have the wild type, the CPVT2, and the FFD, and they are going to look at the frequency of the calcium waves in both group control and with camphorol, and here in control and ruthenium-360. And they will see that in the CPVT2, activation of the MCU decreased the frequency of the waves, and when they inhibit the MCU, it will increase the frequency of the wave. And in the FFD, this is the opposite. By activating the MCU, the frequency will increase, and with the inhibitor, it will decrease. So modulation of the mitochondrial calcium uptake led to divergent effects on calcium waves. And to understand a little bit deeper what was going on, they are going to look at the ROS production, because ROS is known to be produced by mitochondria and to triggers some calcium release by the Rh channels, and so to trigger the other arrhythmias. And they are also going to look at the mitochondrial membrane potential. So first, they are going to look at the ROS production, and so with the camphorol decreased the ROS production in the CPVT2 and increased the ROS production in FFD. So that's maybe the mechanism through which FFD mice go to trigger arrhythmias. And to understand why this, why by activating the MCU, the ROS production of CPVT2 is decreased, they will look at the membrane potential. And why are they going to look at the membrane potential? Because the mitochondria is an organelle really polarized with a rest potential around minus 180 millivolt. And so this encourage the calcium to enter the mitochondria. And so what did they found is that in FFD, the membrane potential decreased really fast, faster than the other groups, which it does not facilitate calcium to enter in the mitochondria. And at the opposite, in wild type and in CPVT2, the membrane potential of the mitochondria is maintained at this potential, which can facilitate calcium to enter in the mitochondria. And here you have the membrane potential after camphorol treatment. And you can see that in CPVT2 is maintained or likely or just a bit bigger, and the time to 50% of the minimal signal is faster in the FFD. So in CPVT2 myocytes, mitochondria seem to play a role of calcium buffer. And as we see just before, in FFD, mitochondria seems to play a role in ROS production. And to determine, to see if mitochondria have other different characteristics, they are going to see on the left with permeabilized cells, the capability of the mitochondria to uptake calcium. And so they've seen that in FFD, it goes faster, but at a lower maximum. And in CPVT2, CPVT2 mitochondria seems to have a bigger maximum and can accumulate much more calcium. And here in intact cells, they are going to pace the cells to reach a steady state. Then they are going to treat them with a tapsigargine, with a SERCA, which is a SERCA inhibitor, to avoid a repump of the calcium in the SR. And they are going to uncouple the mitochondria with the FCCP. And thanks to that, all the calcium of the mitochondria is logged in the sarcolemma. And so as you can see here with the red one is a CPVT2. And you can see that there is a bigger calcium concentration in the mitochondria of the CPVT2. So they've concluded that in CPVT2 and FFD myocytes, CPVT2 and FFD myocytes differ in the intrinsic mitochondrial calcium uptake and accumulation properties. So here they've played with the MCU through pharmacological inhibitors, and now they are going to knock out it. So by knocking out the MCU in CPVT2 mice, the lethality was higher, and also they've seen a severe cardiac remodeling, so hypertrophy of the heart. And they've also seen an exacerbation of ventricular arrhythmias. And in FFD mice, they've just seen ventricular arrhythmias decrease, but slightly. Here they've played with the entering of the calcium in the mitochondria, and now they are going to play with the exit of the calcium from the mitochondria, and they are going to use the CSA inhibitor of the MPTP. Just a quick reminder about the MPTP. This channel have two opening modalities. One permanent, which is well known to be involved in the cell death, and one transient opening modality, which may play a role in mitochondrial calcium extrusion mechanism. And so they are using the cyclosporine A to inhibit MPTP opening, because it is the MPTP structure is not well defined, but it is accepted that the CSA is an inhibitor of the cyclophilin D, which is an activatory protein of the MPTP. So we are inhibiting the MPTP thanks to the cyclosporine A. And so they are going to do the same experiments as with the MCU by loading the cardiomyocytes with cyclosporine A. And what they are going to see is that in CPVT2, by inhibiting the exit of the calcium from the mitochondria, they will increase the frequency of the calcic waves. And in FFD mice, this will not do anything. So yes, we have a calcium wave exacerbation in CPVT2, and also a mitochondrial ROS generation in CPVT2. And so like for the MCU, they are going to make a knockout of the MPTP, and they are going to see that the arrhythmia will be there will be a big increase in arrhythmia in those mice, even if the frequency of the calcium waves will not differ. And to try to understand this, they will measure the mito-wings. So what are the mito-wings? Mito-wings are the transient depolarizations of some mitochondria, which translate, but which when you have a mito-wings is because the MPTP open transiently. And so what they are going to see is that in CPVT2 and in FFD those mito-wings are quite increased, and that the duration of the mito-wings in CPVT2 is decreased. And so to conclude this, this paper, researchers said that in CPVT2 mice, mitochondria play a major role in buffering calcium released during DCR. And in contrast, in FFD mice, mitochondria are vulnerable to mitochondrial calcium overload, which precipitate cardiac remodeling. And so SR mitochondria interplay contributes to the distinct pathologies in the two diseases models by disparately shaping intracellular calcium signaling. So thank you, everybody. And if you have any questions, we can talk about it. I'm going to watch in the discussion.
I have a question, if I can start. From the
Yes, nobody wants to.
choices of the, first of all, thank you. It was frankly quite clear, especially when you took some time to explain the tight connection between the mitochondrial compartment and the sarcoplasmic reticulum, because I worked for years on excitation contraction coupling without taking into account that there is a whole other compartment bound to it. And you show, and the problem of calcium buffering, it's a huge problem for aging, because it's pretty much under the focus that calcium buffering is shifting from the sarcoplasmic, from the organelles, from an interorganellar compartment to the cytosol during aging. And this creates all kind of susceptibility to atrial fibrillation, because or to arrhythmias, even because if calsequestrine doesn't work, calcium is just inside the sarcoplasmic reticulum, and it's interacting with the rear, prompting it to operate. So thank you for that, for taking some time to introduce us to this particular intracellular anatomy. I have one question. You showed experiments with camphorol and ISO, and I didn't understand very well what was the methodology that they used. This one. Yeah. Did they use intact cells, permeabilized fibers? What was the say that the exact protocol? So it was always under ISO.
Yeah, all the time, and after I don't know if it's some intact cells.
Yeah, this is
potential,
maybe
it's intact cells and mitosocs. No, it's TMRA.
Right.
So it's in intact cells.
So tetramethylasteroidamine loading, intact cells, and they do not explain why in some experiments they have control condition and others they have ISO.
Right.
So I suspect
that
for the mitochondrial potential, if they don't put ISO, they have no effect at all.
I see.
Yeah, that's the problem of working on quiescent cells. The ATP turnover is very low.
Sure.
Just to rebound on your question, one big criticism here is that they use the fluorescent probe that only allows them to do quantitative No, it's not
Qualitative.
qualitative.
It's
Okay, so-
mitosox.
Yeah, no, it's tiamari for
Oh,
micro.
yes, sorry. It's
And so you
potassium.
have apps because they do normalize every results they have before CAN-ferro addition.
Uh-huh.
They have no idea of the mitochondrial potential in the individual group of cells.
Uh-huh.
So you have no idea if in wild type CPVT or FFD at baseline, the mitochondrial potential is different. So it's very, very difficult to make any conclusion out of these experiments. And if I would have been reviewer, I would have asked because it's possible to do this kind of experiment to be quantitative on mitochondrial potential.
How does that work, Philippe? How can you do that quantitative?
Oh, so to be quantitative on permeabilized cells, you can use a lipophilic cation that is going to equilibrate from the inner membrane. So, and then you use an electrode sensitive to this cation. Usually you use triphenylphosphonium. It's going to enter into mitochondria, and you have an electrode that is sensitive to TPP in the external medium. And using Nernst equation, you can calculate the equilibrium and determine the true value of potential.
How can you do this in non-permeabilized cells? I wonder.
Not possible.
Oh, no. Okay.
No, as far as I know, no, but maybe there are now on the market some potentiometric dye, ratiometric, potentiometric dye that would allow you to do that. I know that there's one technique using safranin, but the problem is that the distribution of the safranin is really dependent on all the membranes you have on your tissue. And so you have a lot of contamination of your signals. And bear in mind that the SR is as well a polarized organelle within the cell. So it's a little bit difficult. So it's much easier on permeabilized cells.
Okay, thanks.
The second question I have is on the choice that they made in terms of the calcium extruder. So they chose the MTBT, but why not the LCX and CLX?
They've tried to manipulate it, but when they've inhibited, the effect was on both models. And
Okay.
so,
Ah.
yeah, and they've said that it was because it was a major part of calcium exit, and but it would not.
Why not show it? Okay.
Yeah,
well, it's my.
So that's a very good question, Guido. They've been doing so they've done experiment, like Kepa said, the main excretion pathway of calcium within the mitochondrial compartment is the sodium calcium exchanger. And the potential role of PTP into the cardiomyocytes and calcium cycling could be, and I say could, be activated in stress condition. I'll give you only one example. More than 10 years ago, in the lab of Molkentin in the US, they've done a knockout of CIPD. This protein, the only protein known and accepted to be part of the PTP. And when they've done that, the animals, the mice, were quite very well. Impossible to differentiate phenotype of this animal from wild type animal. Two main difference: when you were extracting mitochondria, there were more calcium into these mitochondria. And the second more important point for me is that these animals were unable to adapt to stress. So if you were stressing this animal, they were dying from fatal ventricular arrhythmias, okay? And so they've been showing in this paper that when they are manipulating the sodium calcium exchanger, then a DCR is giving exactly the same phenotype in CPVT and FFD models. But there are more bizarre stuff, because when they are trying to inhibit the sodium calcium exchanger, it's in the supplementary data, they have the opposite effect they observed with MCU. Okay, so when they activate MCU, you increase calcium into mitochondria. When you inhibit the sodium calcium exchanger, you have the same effect. Okay, you increase calcium into mitochondria. So for a same model, CPVT, the same phenotype into the mitochondria, increase of calcium, leads to two different phenotypes in terms of calcium weight or ROS. They don't speak too much about that into the discussion because I think at this time it's not very easy to understand. The take-home message here, I think, is at least for CPVT, they've been, to me, quite interesting argument to demonstrate that this transitory opening clearly is messing up with the electrophysiological stability of the cardiomyocytes. To me, that's a taking home message. In terms of mechanism, I don't really know, but they have two good arguments. They've been using not the ideal concentration of cyclosporine A, but quite a low concentration, so limiting off target. But they have this knockout model of CYP-D that clearly has different effect, but confirming the effect observed with cyclosporine A. So to me, if there's one message to keep and bring home is that possibly for once we have one demonstration that PTP transitory opening is playing a role maybe in electrophysiological stability of a cardiomyocyte in peculiar condition. To me, it's, I don't know, maybe Ruben, tell me if I'm wrong, but to me, a stress condition, so like CPVT, is equivalent to a stress condition for someone that is going, like when you ask an animal or ourselves to increase a lot their heart activity. Okay, and I think PTP action is playing at this point in the under stress condition.
Yeah, yes, I agree. I have a question, and maybe Kepa can answer it, Philippe. What do you think of why they use these two models? Because they're so disparate, they're dissimilar, they differ in many ways that I don't see the value of the comparison. Why did they do that?
They've chosen those two models because they are both characterized by DCR, but I agree that I think there is also many differences, but they've based their reasoning on the fact that they have both DCR.
Yeah. But you know, any model of heart failure shows DCR, I think. So I find it a bit difficult. It reads as if they expect that there is no difference between the two models, so that there is a common mechanism, but apparently there isn't. And I have a second question regarding the electrophysiology. They show some tracings, but for a mouse heart, this is not a tachycardia. And the coupling intervals between the extrasystole and the basic beat is quite high. So how does that explain the arrhythmia and the typical shape of CPVT? Yeah.
Your question is, what are the differences here?
Well, not really, but how does it work? This is not a CPVT, it's not even a VT. You see, for a mouse, this is a normal heart rate.
What is the rationale to call it arrhythmic? Like basically, how is this arrhythmic score calculated?
Oof, I don't really know.
I look at the traces.
There is the baseline, which no, what? It's not really clear.
Well, I can understand that it causes premature beats, certainly, and it does. In the top tracing, you see, premature beats, but I don't see the typical characteristics of a CPVT. So there must be something different in a patient, between a patient and a mouse.
Yeah,
I suspect.
No, it's not even clear looking at these traces because their supplements describe pretty complex protocol to measure ECG.
Injected caffeine.
Yeah, 5 minutes recording baseline injected with epinephrine and caffeine, so intraperitoneally. So two very different mechanisms called into play.
Yeah.
Recorded for another 20 minutes, and it's an arrhythmia score was assigned to each recording based on the time duration of ventricular tachycardia. But and literally, this is everything that is written in the supplements.
Yeah.
Literally everything.
Okay.
So, I don't know. It's the typical score black box.
Yes, yes. I agree.
It's particularly striking, this lethality inhibiting the calcium, the mitochondrial buffering, the potentiality in, in these areas.
Yeah.
Particularly, they start to die at 1 week.
Yeah.
They're not even adults.
No, no, no. Okay.
Also, it would be also interesting to study, but this is more in the context of other pathophysiologies, maybe with more advanced remodeling, the connection between the inositol triphosphate receptor and this kind of mitochondrial buffering, because when there is a lot of ROS production, the inositol triphosphate receptor that is on the sarcoplasmic, on the sarcolemma might come into play more often. And this might explain why the fast food diet, which I suppose is just fructose, right?
Yeah.
It's high fructose. They cannot compensate anymore for more calcium buffering. And this is something that we're looking at also in the arrhythmic model, because inositol triphosphate is very, it's a very to the rescue kind of calcium release signaling.
Hello. Might have some noise because there is some construction building on the moon right
On
now.
the moon?
Yes, it's not American, it's Russian who are coming. I've got a question. If you can put the cartoon summarizing the work. It's always the same question because I'm coming from the EC coupling point of view, and it's bizarre to have some orphan ryanodine receptor like this on the side of the mitochondria instead of at the junctional cleft just in front of the CAV 1.2 L-type calcium channel. So is there any data from super resolution confocal microscopy showing so there is some orphan ryanodine receptor like this just in front of the MCU? And where, on this cartoon, where do you put the circa as well? There is a lag between moon and...
If I might also say, like, this is one of the criticism that I find, because I also, not like you or in detail like you, and I also have studied a lot the excitation contraction coupling. And to me, claiming an arrhythmic mechanism based on some subcellular phenotype without passing to the canonical mechanisms that we believe are pro-arrhythmic, it's a little bit weak. So all the effect that they claim on the ryanodine receptor, they are all based on the literature, which is fine. Just a little bit weak, in my opinion. And so, sorry, please.
But I think, Fabien, the blue moon that you see, that's the SR.
Yeah, but it's not that way. And for me, the sarcolemma or the titubule will be on the other side of the ryanodine receptor, not just in front, because in front there is the mitochondria.
Yeah.
So it's like in a heart failure, the orphelin ryanodine receptor on Philippe, do you know if there is super resolution confocal microscopy or
on these models?
I
don't know if there's any change in anatomy, but yes, you're right. I mean, this kind is just a cartoon, I would say. And yes, I do agree that in front of the type 2 and RYR2, there's the voltage dependent normally, and the mitochondria are adjacent to the cisterna. But then, well, the late modeling would say that there was a radius of calcium diffusion of 250 nanometer around the RYR2. And if you look at electronic microscopy data, they seem to show that mitochondria are sufficiently close to anyway sense the massive increase of calcium coming from the RYR2. And experience in the laboratory that we have done, pharmacologically, when we are destabilizing the RYR2 with FK506. And so we increase heart activity, so the right pressure product. And this is associated to an increase of calcium coming into the mitochondria. Okay, and we see that on the beating heart and on the isolated organelle. So I would say the cartoon is, you're right, not appropriate. Now, I have no idea if these models, so FFD or CPVT, is associated to any change of the ultrastructure of the cardiomyocytes. I have no idea.
Okay.
Ecco,
I've got a second question. If you look at the experiment you showed before like, with isoproterenol and corfeterol, to look at yeah, this one. Why does it take so long for the corfeterol to act? It's only 3-4 minutes.
Yeah, good question.
We don't really know.
I don't know. I've never used that drug, so I don't.
Because I was expecting something like a up.
I don't know the mechanism of this drug.
Okay.
so we'll draft anyway.
And
See you.
also here they are saying that in CPVT2 the capacity of the mitochondria to uptake calcium is much higher, but it takes like 6 minutes to
Exactly. Yeah.
to achieve this, and this is quite long.
But it's if we can go back to the graph before, when the architecture of the sarcolemma, the endoplasmic reticulum and the mitochondria are sense like it makes sense that even if they are not canonically drawn next to each other, the rear and the mitochondrial compartment, because mitochondria canonically are divided by us into the subsarcolemmal family and the interfibrillar family. It's possible that at least one of these subfamilies will be more in contact with the calcium release, with calcium-induced calcium release from the ryanodine receptor, and thus, even because they are anchored to the sarcoplasmic reticulum. So it makes sense that they will be able to sense the calcium-induced calcium release and act on it.
But it's still not clear. I mean, there are many publications debating onto the role of the buffering capacity of mitochondria into cardiomyocytes, because some people have just done very simple experiment where you have an isolated cardiomyocyte, you inhibit the entry point of calcium into the mito, and you have no effect onto the speed of the calcium wave into the ventricular myocytes.
Yeah.
This might be less true for arterial cardiomyocytes. So there's still a debate.
Which compartment is this? Ventricular?
Yep.
Okay. What is MAMS? Is
Mitochondrial associated membrane.
okay.
Okay. Well, you want to say something as a conclusion, Philippe?
Well, I think we have said most of it. I mean, to us, this is very interesting. And I mean, we keep this paper on the desk because I think we have some experiment to finish the Master 2 of KEPA that maybe we'll see if we can, continue into the direction and trying to isolate some of the regulation of this PTP into calcium cycling at the level of mitochondria. What we do know in the lab, which is highly artificial, but ex vivo on the beating heart, if you destabilize RYR2 and that you push the heart to go into an arrhythmia, but burst pacing, you can induce ventricular arrhythmia, stable arrhythmia. And if you do the same protocol in presence of cyclosporine A, then you block this phenomenon. So the heart will go into arrhythmia, but after 3 to 4 seconds, will defibrillate and go into sinus rhythm. And when we've been analyzing, some component into the mitochondria, we've been observing that some stuff that were not supposed to be into the mitochondria and were only supposed to be only into the cytosol were into the mitochondria. And this PTP has a conductance quite important. So every solute under 1,500 Dalton can get through when it opens. So we think that this is happening. Burst has been showing that this is happening on isolated cardiomyocyte, this transitory happening. But for now, nobody knows what is the physiological role of this system, or if it's an artifact, experimental artifact, so we have to carry on. But to me, to see that is encouraging, even if the paper is not perfect, but who can assume that he's making a perfect paper, if it does exist?
Well, it's certainly an interesting paper, if only for the techniques that have been used. I'm impressed by the measurement of mitochondrial calcium and the membrane potential. Very nice. And Kepa, I think you did very well in presenting. It's a very complex paper, and I think that was well done.
Thanks.
Okay, shall we conclude this session then? And then I'll see you on May 9th, I hope. I'll have a serious jet lag at that moment, but we'll see. See you later.
Thank you.
Bye. Thank you.
Bye-bye.
Thank you, everybody.